Okay. So today's Wednesday, exam on Friday. I posted a Word document on a little bit of tips and strategies into EduCat in the PowerPoint section, where I post all the PowerPoints. Did you see it there? Did someone look for it? Okay. So it's there. I can leave a few minutes at the end of this lecture period. If you have any questions regarding the exam content, if there's anything you need clarification on, I can do that. But on Friday, make sure that you come prepared to do Respondus lockdown type exam and make sure your battery's charged up. And then Jackie will be here to help out so that I can have an extra set of eyes. I'm hoping that if you have questions, you'll be raising your hand and I can come over and help you. And then when you are all done, make sure to either check with Jackie or myself that and show us that you submitted both versions of the exam, the multiple choice and the essay. Okay. Before you leave the room, you need to do that. Questions right now on the exam, like procedure or anything? I think we don't have anyone coming in after us on Fridays, so if you don't have to immediately jet off to something else, you could stay a little bit beyond 2:50. Okay. All right. Well, I wanted to continue on with part 2 of cell structure and function and look at some of the unusual or unique structures that some microbes could have. And topping the list is probably the idea of a flagella and how they use that to get from point A to point B in a directed manner. And so flagella, if you're just talking about one, you say flagellum, and it is a complicated structure, way more complicated. When they started looking at it more carefully, there's lots of parts to it. So I'm going to list out the parts and then I'll show you a picture and put it all together on how it works. And I'm guessing most of you are more familiar with flagella in animals, eukaryotic cells, like sperm flagella, for example. You've probably seen video of that where the flagella seems to have like that whip-like motion to it. And that's not how flagella work with bacteria. They don't whip, they rotate. So they're rigid structures that will rotate, kind of like a motor propeller on a boat, if you want to think of it that way. So there's actually a portion that they call the motor portion. And these are proteins that will be anchored in the cell membrane. And then you'll see that it's composed of this, what they call a ring system. And then, so it's like a ring you wear on your finger. So that kind of idea. And then through that ring will be the rod. So they call it the central rod because it comes through the ring system. And there's going to be several rings. It depends on which bacteria you're thinking about, how they assemble their rings. And then there's this so-called MOT proteins. I say MOT, probably some people say MOT, but I say MOT proteins associated with the so-called motor portion. And what they're finding with those MOT proteins is that's what's actually going to allow for the rotational force or the torque force that will allow for that rotation for the flagella that move through space. So the filament, by the way, is the actual thing that you would see sticking out of the cell. So a lot of people call that the flagella, but it's really the filament of the flagella system because the flagella is actually a complicated bunch of bits and pieces. The flea proteins are another idea that are associated with the flagella, and what it allows, what they think, it seems to allow the flagella to rotate in the opposite direction. So maybe at first the flagella will be going counterclockwise. And then the free protein kick in and then allow that flagella to switch and go clockwise. And so it's this switch between counterclockwise and clockwise. And it seems like the free proteins are necessary for that. Okay. So pretty much all we've just described here is pretty closely associated with the cell membrane and is in that region. And then we see that, there's this central rod that comes through, and then that connects to the hook section. I'll show you a picture. And the hook, think of the hook as like the intermediary. It's the piece that allows the central rod and the filament to actually kind of connect well. And so the hook needs to be there to facilitate the attachment of the filament, essentially. And that filament, that's the long, thin part that you see sticking out of the cell. And again, it's not a whip-like flexible thing. It's rigid. It gets built into kind of a helical or corkscrew pattern of building it, and then it stays rigid. So the filament, when they've looked at it carefully, it's just all made out of protein subunits that get made inside the cell, and then they get sent out, and they kind of like just keep building and building and building to the length of the filament until it reaches its final length. And again, it's going to be in an overall helical shape and rigid. So let me show you a picture of all of this that I just put in words is being demonstrated here in this image. This is not from your book. It's from a different microbiology book. I don't know, I like this image a little bit better. I'll show you the one in your book as well, but this one's a little bit bigger, a little bit more obvious. All right. So we see the cell membrane. Okay, so let's look at this a little carefully here. So here's your cell membrane down here. And then you see this really thin peptidoglycan. And then you see this outer membrane. So what kind of cell type are we looking at here? This is gram-negative. Okay, gram-negative cell type. So again, they know more about the E. coli. E. coli's gram-negative. They know more about E. coli's flagella probably than any other bacteria. So E. coli tends to be the guinea pig for studying this. All right, so here you can see those ring systems, and you can see that there's, in this particular case, they're showing 4 different rings. They're different colors. You don't need to know the letter, just know that there's rings involved, okay? And then the rod goes right through those rings and then connects to the hook. And then the hook connects to the filament, and all those brown tannish dots, those are just the protein subunits that get built and then sent through the hook and added on. So it keeps adding on and adding on and adding on until it reaches its final length. Then you see the MOT proteins and the flea proteins down in the cell membrane area. Okay, so they're associated with each other. They're making physical contact. So what we see happening is they are showing off to the right here how to get this flagella to actually rotate. So here they're showing 2 of the rings, the C and MS, so they're focusing in only on the 2 bottom rings. And then they're zooming into the MOT proteins. And you see that there's protons flowing through the MOT proteins. And that's actually what's allowing for the torque force. So what they're implying here is that the bacteria has to create a proton gradient. So what it's doing is it's going to have to create a situation where there's lots of protons building up outside of its cell, across that cell membrane. There's fewer protons inside the cell, and so then you get this proton gradient. The protons are going to want to, chemically speaking, they want to even out across the membrane. So through the laws of diffusion, they can't go right through the membrane because that's too hydrophobic. They're going to have to figure out a way to get through. They do this in the MOT protein. So the MOT proteins facilitate the movement of the protons back into the cell. And as they're flowing through the MOT protein, that actually creates a situation that generates that torque force. And so as they're flowing through, that torque force happens and you get that rotation of the flagella. And so that gets transmitted all the way out to the filament, you get that rotation. So that purple swooping line that you see up top here, they're trying to show, in this case, that the flagella looks like it's going counterclockwise, okay? So lots of parts that need to get built, put together in the right way, and then you have to use a significant proton gradient. So I have in my notes that one group of scientists, when they were looking at how many protons does it take to get a rotation, their studies showed 1,000. 1,000 protons have to come through to do one rotation. To me, that sounds like a lot. And so I'm going to double check that and see if there's been any updated studies done, because that's phenomenal. If that's truly the case, that's a lot of energy to devote to this. And so they have also studied, like, what does it take to build this thing? You know, so you can imagine that all these different proteins, those are different genes on the chromosome that have to get turned on. And then you get, you know, transcription, translation, build the protein, get it assembled. And when they've looked at that, the earlier studies, and I'm going to update and see if they've come out with anything newer. But the most recent study I found, 40 genes are necessary to get turned on in order to build. And then they're claiming 1,000 protons to go through the MOT protein to get a rotation. So, yikes, that's a lot, right? So please look over. I think this is a pretty good diagram because it nicely shows all the different parts. And be able, not on the exam on Friday, but on the next exam, be comfortable talking about the flagella or recognizing questions about the flagella, like how does it get put together? How does everything connect? And how does it move? Because that's pretty significant way for bacteria, you know, to be able to move in a directed manner. Your book shows this one, which is good. Basically the same thing. It's just not as big, but pretty much everything is the same. So if you like this one, that's fine to study off of as well. Questions on the flagella? Flagellum?
So, these two models are showing the gram-negative bacteria, or their flagella,
Yeah. And they're less complicated. This is more complicated because you have to do extra rings into the outer membrane in particular. So, you know, to be honest, I have not seen this level of detail for gram-positive flagella. They seem E. coli, a lot of pathogens that attack us are gram-negatives, and so they tend to get the limelight. You know, historically, they've been studied the longest. Other questions on the flagella? Well, let's think about movement. So here you're looking at a couple of different ways that they've studied the ways that the flagella gets used by different bacteria. And you can see sometimes they have more than one flagella. Can you imagine having to build more than one? It just seems like a lot of work. So the top image, you're seeing many flagella, and they say they get bundled together. It's bizarre. So they will, what they mean by that is the flagella kind of lay down next to each other and kind of get in sync with each other, and they'll rotate in sync. And that top image, you can see that it's, in this case, they're saying it goes counterclockwise. I guess it'd be like that. It's going counterclockwise. And then you can see that at some point, maybe the bacteria senses it needs to shift direction. And so it will actually cause the flagella to fly away from each other, move away from each other very abruptly, and it stops the cell. They'll stop moving, and then they can reorient. And sometimes it's just through like getting hit by water molecules that it causes them to kind of reorient. And then they'll bundle their flagella and move on. And you can see this under the microscope. If you look carefully, you can see them making this steady progress, and then all of a sudden they seem to like stutter and kind of wiggle in place, and then they start to move off again. And so bundling their flagella is a strategy. So I would like you to know this word here, paratrichus. So if they didn't do a great job, I'm going to well, the next image shows a little bit better what paratrichus is. But when you say paratrichus, it's usually a rod-shaped cell, and they have flagella all over, not just at one end. They're like embedded all around the cell. And it's not showing the best. Then you see in the middle image reversible flagella. So here they're showing that you can push the cell by rotating the flagella. In this case, they're saying counterclockwise, and then you can pull the cell if you reverse the rotation. And that's an option. With some bacteria, we'll do it that way. And then the bottom image, unidirectional flagella, is again this idea that they only seem to really go, in this case, they're showing clockwise, and then the cell will stop moving its flagella and reorient passively and then start up again, right? And so we do see that. And when you see, well, do I have it? Okay, here. So when you see that kind of movement, you would call polar microbes tend to do this. So when we say, oh, look at that, it's a polar flagella, what we mean is that it's just one flagella, usually on the short end of a rod, okay, like on the end of the rod. Then we say it's a polar flagella. So let's look at this image of actual real bacteria where they did a very fancy flagella stain. Flagella stains are really hard to do. So I've purchased prepared slides that you'll see in lab because I've had students in the past try the flagella stain, and it gets really frustrating. Like no one can get it to happen. It's really frustrating. It's just a really tough staining technique because you have to, flagella are very fragile. And then those of you who know what mascara is, that is essentially what you're doing. You have to coat the flagella with like layers of the dye, kind of like you do with eyelashes and mascara. And it can be quite challenging. But figure A is showing paratrichus. So they're trying to show how the flagella are attached all the way around the cell. So that's E. coli. E. coli is like that. Then polars in the middle, just one flagella on one end. And then I'd like you to know that last term, Lophotrichus. Figure C is showing lopho. That's a tuft of flagella, but just on one end. A tuft of flagella, just on one end. So usually what we see with Lophotrichus, and pretty much with Paratrichus as well, when you have lots of flagella, when you watch them under the microscope, they tend to move slow and steady. Like they just kind of like a big ocean liner. They're just really slow and steady moving up through your field of view. On the other hand, if you look at a polar, a bacteria under the microscope that has a polar flagella, they tend to look like they drank a whole pot of coffee. And they're zooming around really quick and they change direction pretty quick. And so just by observing their movement speed and kind of how chaotic they might be, you can actually tell what kind of flagella they have within good reason. So it's kind of fun to watch under the microscope. Okay, here's another example of polar up top, and that's a really good one of Paratrichus on the bottom there. So can you imagine having to build all those? That's a lot. Yes. It's bizarre. It's like why? I mean, it kind of makes you go, why? Why would you do this? And I don't know. I don't have an easy answer for that. What kind of selection pressure does it have where it felt it needed to, through an adaptation to its environment, make all these flagella? I don't know. Unless they're using the flagella for other things besides movement, maybe then it would make good sense.
Would it make sense to be an
Yes. Or having a way to really quickly generate a proton gradient. So yes. And
um,
yes.
if they run out of if it's less photons, we stop.
Yep. They're stuck. They're stuck in stationary. So, and then the other thing too, which I've struggled with in teaching this class in lab. Okay, so let's say you're E. coli. E. coli looks like the lower one. It has paratrichus. So now the E. coli we work with, when we grow them for you and I store them in the deep freeze for you, and they get everything they want. I feed them all the luscious ingredients, all the nutrients they want. They get nice warm temperatures. They don't swim around in their waste, so they're not poisoned. I give them everything they need. They're baby. If you have that kind of life, would you build all those? Okay, so I'm creating a selection pressure, not on purpose. It's just the way that we work with microbes that's, efficient and quick. And so some of the times when I ask you to do, like, we're going to do a motility test coming up pretty soon, actually. And I've had it fail because I didn't realize, like, we gave you E. coli that said, I'm not going to do this. I got the sweet life. I'm not doing this. So be aware that sometimes in lab conditions where that microbe has been baby, they might forfeit this, which makes sense, right? From their perspective, why bother? And then the other thing too, sometimes they won't build it, or sometimes they build it, but it won't, they choose not to use it. Like even if there is a proton gradient, they can direct that proton gradient for other uses, because microbes use proton gradients for a lot of different reasons. And so they might just not put it through the mock protein. So again, but there have been actual studies where they can show E. coli evolved to stop making flagella, like the lab E. coli, the ones that are used in labs across, the different universities, research facilities. So, okay, so flagella is very good strategy if you're in an environment where being mortal really is a payoff. Go ahead and do that because it can be very quick. It can get you out of dangerous situations or into good situations very quickly. And so you might want to move toward a source of nutrition, for example, if you're sensing great, patch of nutrients, you might want to move toward that very quickly and get there first. If toxins are in your environment, you can easily swim away, so to speak. But you need to be, suspended in some form of liquid. Now, you have to remember how tiny microbes are. So you might only need a thin film of liquid in order to use your flagella. But you need some form of liquid to rotate that flagella through it, just like a motor in a boat. This gliding motility is another strategy that they've seen, and they don't know as much about it. And there's still a lot of mystery to it, but they know flagella don't seem to be involved in this. And it's actually some protein, special proteins that are embedded in the cell membrane and kind of stick up off the membrane out into the environment. When you watch gliding motility, it's slow, very slow, but it does get them from point A to point B. And it's usually a rod-shaped cell that they've noted this in. So across the long axis, most rod shapes, you have that long axis. And they don't do it if they're suspended in water or a liquid. They won't do a gliding. They have to have physical contact with something solid. All right, so like the agar surface, the plates that you'll streak bacteria out on, every once in a while you might find that one of them will glide across the agar if you let it incubate long enough. So they're still investigating this. They can see that it seems some bacteria do this just by secreting out a slime, a polysaccharide slime, that if they do it in the right way, it allows them to kind of move forward very slowly. And Some do it through the use of pili, which we've talked about a sex pilus for conjugation, but there are other types of pili. They usually number them Roman numerals. This is the type 4 pilus. And when they've looked at this more carefully, what they think is going on is that, again, the tip of the pilus kind of reaches out, so to speak, and the tip touches the surface, and then it kind of ratchets the cell forward. And so it's kind of like when you climb a wall. What is that thing that when you throw the hook over the wall and then you climb up the wall? What am I thinking about? What is that? Grappling hook. Yeah, thank you. The grappling hook idea is kind of like that, where the tip of the pilus is kind of like the grappling hook, and then you can pull the cell toward that. And then probably the more well-studied is proteins that are embedded in the cell membrane, and they make contact with the physical surface. And again, they're not quite sure exactly how the movement occurs. Let me show you a picture here where the mock-up is. Again, they're showing a proton gradient might be needed, but again, that's still a little murky. They have strong suspicions that a proton gradient is needed to generate the force necessary to get the proteins to do their job. So some papers I've read, they made it sound like, you know, the proteins will move forward a little bit, but they're in the membrane. They don't let it come loose. They just kind of move forward a little bit and ratchet the cell forward. And but again, very slow movement compared to when you look at flagellum and how fast those cells come. So gliding movement is a thing. I'll show you some pictures. So here you're looking at on the left, this is a Flavobacterium genus of bacteria. And here, what they did in both cases, in both images, is they spot inoculated, which means they put dots of cells in the center of these images and then let them grow. And you can see the one on the left, how it's, you know, so it was probably spot inoculated, like right there. And then it grew out from that spot. You know, you can see how you actually have kind of long, slender movements here that are coming out. And then the ones on the right are 2 colonies that are have been made mutants to no longer have the ability to glide. And you can see the colonies stay tight and there's no movement away. I've often seen gliding motility in bacteria that I isolate from the soil. So this is a soil dilution that I did in one of the experiments we were running. And you can see there's several interesting colony types here. So I'm zooming into this region up top. I'll zoom into that in a second, but you can kind of see some movement there. I'll show, but let me, before I do that, all of these cells here that you see, that these huge cells, and they kind of look like hairs, they look hairy. That is very common from soil samples. Those are a bacillus, most likely a bacillus genus of bacteria. And they're famous for doing that kind of hair-like curly growth pattern. And so they are moving out as well. But here, the ones on the top, you can kind of see the path, you know, that hazy path behind the colony, so to speak. And so they're moving forward en masse all together. And so sometimes we see that behavior too, where many cells will coordinate their movement and move as a team across the agar. And they think it's to like go access food. And sometimes, well, they're finding more and more now that bacteria that live in like more natural conditions, they often will team up to like go find food. Like, so for example, if they come across a patch of nutrients that maybe is a polymer, you know, something very complex. And what if that one bacteria doesn't have all the enzymes necessary to break it down, but a neighbor does? And so often they'll coordinate their efforts and they'll like team up to like, well, I have this enzyme, I can break it down to this substrate, then you use your enzyme and break it down even further, and then we can all enjoy the feast. And that's kind of what happens. And I'll talk a little bit more about that. And so we're realizing that microbes, you know, they can team up like this and cooperate for different reasons. here's a beautiful one. I just thought this was done on purpose to try to create, what they did was they spot inoculated right here. And then they grew it across an aquifer that was devoid of nutrients. So they were creating a situation where they're forcing the microbe to keep moving. And it created these very beautiful patterns. You can have fun with that if you find a microbe that can glide or move in some way like that and create these really cool patterns. Okay, why don't we try an iClicker? I gotta ask you a question.